good afternoon everyone. Today I explain classification of methods of a color measurement or comparison. This basic principle of colorimetric measurements is to compare the color produced by substance in an unknown amount, the sum color produced by a known amount of the material beginning determine the quantitative comparison of this solution may be carried out by six methods. Okay, first method, standard series method, duplication method, dilution method, and balancing methods. And fifth, photoelectric photometers, spectrophotometer methods. This basic principle of colorimetric measurement is compared to the color produced by substance in an unknown amount and with the same color produced by known the amount material beginning determined. Okay, ह्या मेथडमध्ये सगळ्या मेथडमध्ये काही नोन अमाऊंट ऑफ द सॅम्पल्स प्रोड्यूस केल्यानंतर आपल्याला अननोन सॅम्पल हे डिटरमाइन होत आहे. ओके, अँड वी विल डिस्कस दी टू मेथड्स हिअर, photoelectric photometer and spectrophotometer methods. First of all, photoelectric colorimeter methods, that is a single beam photoelectric colorimeters. And the photoelectric colorimeters may be divided into two main classes, that is a one cell and two cell instrument. Let us consider here only one single cell that is single beams and filter photoelectric photometer calorimeter, and this essentially parts, that is light sources, light up filters, and photocells and barrier layer photocells, container for the solution, and last the galvanometers. Okay, photoelectric colorimeter, there are the two types, single one cells या single cells and two cells, that is a double beam of photoelectric photometers. आणि त्याचे काही सोर्सेस आहेत, कशाचे, फोटोइलेक्ट्रिक सिंगल सेल्स फोटोइलेक्ट्रिक फोटोमीटरचे. त्यामधले फर्स्ट सोर्सेस होता ए लाईट ऑफ सोर्स, ए लाईट ऑफ फिल्टर, and third source is a container for the solution, and fourth a barrier layer photocells, and fifth a galvanometer. Okay, a light of sources means sunlight, arc light, mercury vapor lamp or discharge tube is used for this purpose, and here we can use 2 to 6 volt lamp is operated, and constant voltage is transformer. A light of sources मध्ये sunlight, arc light, mercury and vapor lamp या discharge tube is used for this purpose. लाईट सोर्सेस म्हणून सनलाईट किंवा आर्क लाईट या मर्क्युरी वेपर लॅम्प्स डिस्चार्ज ट्यूब्स यूज केले जाते, आणि त्याचे एक कॉन्स्टंट व्होल्टेज मेंटेन केलं जाते. ते किती, तर 2 to 6 हे कॉन्स्टंट व्होल्टेज लाईट ऑफ सोर्ससाठी असतं. त्यानंतर सेकंड ए लाईट फिल्टर, दॅट इज अ मोनोक्रोमॅटर. ओके, दिस इज कन्सिस्ट ऑफ ए फिल्टर or a prism in a spectrograph, and light of a suitable wavelength coming out of the monochromator is selected. A filter consists of colored glass or colored gelatin coated on a glass and possesses this property of a transmitting high from a specified region of the spectrum. Okay, and this ideal way of selecting a filter is to construct the first absorption curve for the visible spectrum, and here is a optical density or concentration calibration curve may be constructed with a photoelectric colorimeter, and using each of filter is supplied is turned, and this base filter of this which gives the maximum absorption or minimum transmission for a given concentration of absorption substance, and this complementary colors by this wavelength in nanometers. Okay, a light of filter, and that is a monochrome matters. It consists of filter prism spectrograph. ह्याच्यामध्ये काय दिलेत, वेगवेगळे तुमचे कॉन्सन्ट्रेशनचे सोल्युशन असतील ते यूज केल्यानंतर सॅम्पल्स म्हणून जे मॅक्झिमम सॅम्पल्स असतील त्या मॅक्झिमम सॅम्पलचा तुम्ही प्रत्येक फिल्टर्स बाय चेंज करत गेल्यानंतर जिथं मॅक्झिमम रिझल्ट येईल वेव्हलेंथ, आणि तोच काय असेल तुमची ऑप्टिकल डेन्सिटी जिथं मॅक्झिमम आलेले असेल ते कॉन्स्टंट ठेवून तोच त्याच फिल्टर नंतर प्रत्येक सोल्युशन कलरचे जे कॉन्सन्ट्रेशनचे असतील त्यांची ॲब्सॉर्बन्स मेजर केला जातो. मग या ठिकाणी काय प्रत्येक कलरच्या प्रत्येक कलरवरती त्याची काय वेव्हलेंथ मेजर केलेली आहे कॉन्स्टंट नॅनोमीटरची. ते फिल्टरवर प्रत्येक कलरचे वेगवेगळे आहेत ते कोणते, तर इन दिस व्हायलेट कलर्स देर इज 400 to 435 कलर्स नॅनोमीटर्स वेव्हलेंथ, अँड ब्ल्यूज 435 to 480, yellowish green 500 to 60 and to 580, and number of colors, and there is a ranges in this chart. Okay, complementary colors of the chart. And third sources, a container for this solution or cells or cuvettes, and these cells or cuvettes are rectangular or cylindrical vessels with the accurate dimension. If this visible spectral range is used, glass vessel can be employed, and however it is ultraviolet light is used, and these vessels or these optical parts and systems must be made of quartz, and this working of surface of the cells should be very clean, and this measure the made with the cell empty or filled with the pure solvents and then filled with the solution under this study, and difference between these two measurement and given the light radiation is absorbed is container for this solution. याच्यामध्ये स्टार्टला तुम्ही काय करणार आहे, इन्स्ट्रुमेंट वर्किंग करताना ते दिलेली कुवेट्स असेल ती कम्प्लिटली क्लीन या ड्राय करणार, त्यानंतर त्याच्यामध्ये एम टीची कुवेट फिल करून सो ॲब्सॉर्बन्स चेक केला जाईल, त्यानंतर फिल दिस सोल्युशन and there under this study. Okay, and difference between the two measurements and given light radiation is absorbed. Okay, and कुवेट सेल असेल त्याचं कसं एक ठराविक शेप असतो, एक रेक्टँग्युलर किंवा सिलेंड्रिकल, आणि विजिबल स्पेक्ट्रल रेंज इज युज्ड, ग्लास वेसल कॅन बी एम्प्लॉयड, ग्लास त्यातून रेंज पास झाली पाहिजे अशा टाईपचे कुवेट्स किंवा सेल्स असतात, आणि त्याचा शेप रेक्टँग्युलर या सिलेंड्रिकल फॉर्ममध्ये. ओके, नेक्स्ट फोर्थ टाईप पार्ट्स, ए बॅरियर लेअर फोटो सेल्स, दॅट इज अ फोटो व्होल्टॅटिक और फोटो इलेक्ट्रॉनिक सेल. This is one of three photoelectronic cells, namely photoemissive, photoconductive, and photovoltaic cells, and these cells can be represented as it consists of an iron metal base plate is shown as the A. Metal base plate is shown as the A, and on this which thin layer of selenium B is deposited, and this is covered by the transparent gold metal layer is denoted by D, which is varnished, accepted, and portional mechanically strengthened to the form consulating ring is denoted by E, and this underline of this metal base plate is covered by a non-oxidizing metal, and when light is passing through the thin metal layer and falls upon the selenium surface, electron and related which penetrate and hypothetical barriers layers is denoted by C, and given transparent metal layer is denoted by D, and negative charge, that is a collecting ring, is denoted by E, acquires as a negative polarity, and while this metal base plate acquires in positive polarity, and cell will be formed, and which is give connected to galvanometer and will be give flow current, and which vary will intensity and incident light, and photovoltaic cell is offered and without the use of battery, and they are highly sensitive and more than these photoemissive cells, and they are portable and not more costly, and photovoltaic cells exhibit and fuse an effect and have considerable temperature of coefficient. फोटोइलेक्ट्रिक सेलमध्ये काय झालं जाते तर सगळे लाईट ॲब्सॉर्ब जिथून काही आलेले असतील ते इमिशन लाईट ॲब्सॉर्ब केले जाते, आणि त्याच्यानंतर जे काही हे असतील लाईट्स वेवलेंथ रेंजेस ते पास गॅलनोमीटरमध्ये केले जातो. नेक्स्ट fifth part, galvanometer and is device for measuring and response of photocells, and in a photoelectric colorimeter this response from the photocell and is measured by means galvanometer, and which gives this correspond deflection, and following the figure gives this schematic representation single photoelectric colorimeter. In source of light is transmitted in collimating lens and transmitted to filter and movable holder reaction of cells या cuvette and transport to photo cells, and photo cells transmitting to galvanometer, and galvanometer is a record to optical density. Here the light Here the light from source passes through collimating lens and in parallel beam often then passes through suitable filter to give light particular wavelength, and this light passes through solvent solution and plays the cuvette, and this radiation transmitted by solvent revised by the photocell which produce this deflection of galvanometer and connected it and determine the unknown concentration of galvanometer readings are taken say the standard solution of a given substance, and which increasing concentration by using pure solvent and refers, and these readings are plotted against concentration to get calibration curve as shown this figure, and using calibration curve is unknown concentration can be determined. कॅल्ब्रेशन कर्वमुळे काय अननोन कॉन्सन्ट्रेशन डिटरमाइन होते. याच्यामध्ये गॅलनोमीटरमध्ये काही रिडिंग्स फाइंड आउट झालेले आहेत. ते रिडिंग्स काय वाय ॲक्सिसवरती मेजर केले, आणि कॉन्सन्ट्रेशन डिफरंट कॉन्सन्ट्रेशनचे काही सॅम्पल ऑफ सोल्युशन कुवेट्समध्ये घालून वेगवेगळे त्यांचे रीडिंग घेतली होती. ते एक्स ॲक्सिसवर असणार आहेत, आणि त्याच्यावरून डिटरमाइन काय होणार, नोन सॅम्पल सोल्युशनवरून अननोन सॅम्पल सोल्युशन डिटरमाइन होणार आहे, and that is calibration curve is known as a unknown sample solution result. Okay, next method is a spectrophotometer method, single beam direct reading spectrophotometer, and this is a most accurate method for determination of the concentration of substances in solution, but these instruments are more costly, and this spectrometer may be regarded in refined filter photoelectric photometer, which permits use of continuously variable and nearly monochromatic bands of light. And these essential parts spectrophoton are the source of radiant energy, and source a monochromator, and glass or silica cells, and device of receive measure of beam's radiant energy passing through the solvent or solution. And this spectrometer method is mostly accurate method, and this determine for concentration substance of the solution, and but these instruments are more costly. ओके, जास्तीत जास्त कॉस्टली इन्स्ट्रुमेंट आहे. आणि स्पेक्ट्रोफोटोमीटर दॅट इज अ सिंगल बीम डायरेक्ट रीडिंग स्पेक्ट्रोमीटर, ज्यामध्ये डिजिटल या डायरेक्ट असे काही तुमचे रीडिंग्स असतील ते सॅम्पलचे मिळतात. Instrument layout, the working, this construction of layout. Single beam spectrophotometer is shown in this figure. In image of the light, A is mounted on the movable form focused by considering A, and A is a light sources mounted on the movable arm unfocused by considering mirror, and B is a diagonal mirror, and C on this entrance slit, and D is a light filling on the collimating mirror. E is a parallel and reflected to the quartz prism F, and the back surface of the prism is aluminized, and so this light is refracted as the first surface and reflected back through the prism, and undergoing further reflection and immersion from this prism. And this collimating mirror focus prism placed in the slit is denoted by D, and light of this selected to the absorption cells as the G, and then this photo cell is denoted by H capital, and this response of this which amplified and this noted, and this resisted on the meter and is denoted by capital M. In fact of this visible and nearly ultraviolet part of the spectrum, and deuterium lamp for this ultraviolet region. In similarly there is the two phototubes, and from which appropriate phototube is brought, and focal point of the lamp are changed over this in modern instrument. And this prism F will be replaced by diffraction of grating. Colorful this spectrophotometer methods in diagram. Light sources and collecting lens is absorbed and transmitted to monochromator prism grating. In wavelength is selector of a slit and sample solution of cuvette transmit to detector of photocells and digital display of meters. Last point of this topic, application of colorimetry and spectrophotometry. There are the application of colorimetry. This colorimetry is commonly used for the determination of the concentration of colored compound by measuring the optical density or absorbance. The colorimeter is commonly used for the determination of concentration of colored compound by measuring optical density or absorbance. कलरीमेट्रीचा यूज जास्तीत जास्त कलर ऑफ कंपाऊंड्स काही सॅम्पल असतील कॉन्सन्ट्रेशनचे वेगवेगळे त्यांच्यामधलं मेजर्स ऑप्टिकल डेन्सिटी आणि अब्सॉर्बन्स मेजर किंवा डिटरमाइन करण्यासाठी यूज होतो. त्यानंतर कलरीमेट्री कॅन बी यूज्ड फॉर दिस डिटर्मिनेशन ऑफ द सोर्स ऑफ रिॲक्शन बाय मेजरिंग द रेट ऑफ फॉर्मेशन अँड डिसअपिअरन्स ऑफ द लाईट absorbing compound in the range of visible spectrum of light. Visible spectrum of light वेगवेगळ्या कॉन्सन्ट्रेशनचे सॅम्पल्स यूज करताना कलरीमेट्रीचे यूज केला जातो विजिबल ऑफ लाईट. अँड बाय यूजिंग कलरीमीटर कंपाऊंड कॅन बी आयडेंटिफाय डिटरमाइन दी ॲब्सॉर्प्शन स्पेक्ट्रम इन दी विजिबल स्पेक्ट्रम अँड इट कॅन बी यूज्ड टू मेजर अँड दिस concentration of chiral concentration and intensity of variety of material in food irrigations, building materials, textile products, beverage and chemical solution. हे बिल्डिंग्स मटेरियल्स असतील किंवा टेक्स्टाईल प्रोडक्ट्स काय असतील या फूड इरिजेंट्स अदर्स काही मटेरियल्स असतील त्यांची कॉन्सन्ट्रेशन मेजर करण्यासाठी यूज कलरीमेट्रीचा केला जातो. त्यानंतर इट इज वाईडली यूज्ड इन हॉस्पिटल्स अँड लॅबोरेटरीज फॉर इस्टिमेशन्स ऑफ बायोकेमिकल सॅम्पल्स लाईक प्लाझ्मा, सिरम, युरिन, cerebro hospital fluid that is used widely used in hospitals, yeah, laboratories. Okay, used for this colorimetry. It is used to determination amount of glucose, urea, creatine and lipids, total proteins and minerals, and biochemical samples like plasma, blood, urine, serums in concentration used to the colorimeters. And their next application of spectrophotometry is used in molecular biology and measuring the growth of microorganisms like bacteria. And spectrophotometry is used in forensic science. And spectrophotometry is used in a determination of molecular weight of molecule. Okay, and fourth application, spectrophotometry is used in unknown concentration of the sample solution, and it is used in characterization of aromatic compound and is detection of composition. And sixth application, it is used in determination of impurities present in the samples. And UV visible spectroscopy is mostly used in pharmaceutical, cosmetic industry, analysis of additive and coloring materials. In food analysis, particularly analysis of wine, spirits, other drinks, fruit juice, meat products, milk and dairy products, oils, sugars, vegetable products, and agricultural supplies, analysis of animals, feeds, effluent controls and analysis herbicides and pesticides, and analysis of blood plasma and formed elements in the blood cells. The application of three types of application discuss here: application of colorimeter, application of spectrophotometer, and application of UV visible spectrophotometer is a use the today I discuss the two methods, classification of the two methods in photoelectric calorimeters and spectrophotometers, and their sources discuss.